Okay, so, oh my goodness. All right, good luck on whatever's going on there. So again, we're going to be talking about lipids, biological membranes in this chapter. And right here we can actually see some nice biological membrane action going on, as it were. Okay, so again, the fatty acids are going to be your key constituents of your lipids. The lipids are going to be the water-insoluble biomolecules that are highly soluble in organic solvents. Notice the lipid definition in your biochemistry textbook is very intentionally broad, okay? There's actually a very wide variety of lipids, right? Though fatty acids has a much narrower definition, which is that fatty acids are going to be long hydrocarbon chains that terminate with carboxylic acid groups. So note that lipids is a very broad definition that has a large box, right? And this is going to mostly be hydrophobic due to fatty acids. But, again, lipids are a very broad box, okay? The fatty acids, on the other hand, are going to vary in length and degree of saturation. They're often going to be referred to in their carboxylate form because they're ionized at physiological pH, and essentially you would never expect to find a fatty acid in a living organism at anything that's not carboxylate, okay? They essentially will never be either carboxylic acid form because it just does not get that low in pH in most organisms. But you can. The fatty acid names are going to be based on their parent hydrocarbons. So the fatty acid name is derived from your parent hydrocarbon by the substitution of oic, literally the oic form, ic acid, because carboxylic acid, right? Okay. The names are going to be things like octadecanoic acid. The first number in the number is the number of carbon atoms, and your second number is the number of double bonds. And so right here we have palmitate. This is the common name for that chain. This would be palmitic acid, which is again the derivation of the comic name for the carboxylic acid group right here. Okay? Fantastic. Does anybody have any questions about that so far? Okay. If you make this really, you put an ion here, like a sodium or a potassium or something like that, a +1 charge ion, and you put these things with a little bit of base to help them make sure they go all to that form. Do you know what you call that? That's soap. Why you can make cells and fats from animals and put them with a strong base and make soap, right? Okay. Okay, so carbon can be numbered starting at the carboxyl terminal carbon atom. The carbon atoms 2 and 3 are often referred to as the alpha and beta, respectively. And again, that's this right here, right? 1, 2, 3, alpha, beta. Everybody remember that from organic chemistry? Fantastic. So, the position of your double bond can be represented by a symbol of change, delta, followed by the superscript number. So, for example, we would have a cis double bond. It would be at delta 9. We have a trans double bond. It's at delta 2 in this case. All right. So the methyl carbon atom at the distal end of the chain is going to be called the omega carbon atom. You know, alpha and omega. You've probably heard of that since we're a Christian university, I hope. If not, we got problems. We have a lot of problems. Yeah. Yes, that's the number down on the chain. Starts. Yep. Double bonds are always defined by starting. Okay. So omega, that's the last carbon in the chain. And so that's going to be the distal end of your chain, the omega carbon. Omega is this symbol right here, by the way, that looks a bit like a W. So the position of a double bond, this can be represented by counting from the distal end it's going to be a W3 fatty acid is going to be shown down here. And so again, here we have the omega, and then 1, 2, 3. These right here are the methylene groups, by the way. Remember that when you have a double bond, you're going to end up with a methylene group, which means one less hydrogen, right? Okay, so the methylene groups are the normal groups that you would see for a chain. The CH groups will be the ones in the double bonds, and that will be degrees of unsaturation, just like we learned in organic chemistry. Okay, so this right here, again, this is a alpha beta right here, right? That's the second and third carbons. This right here is the delta 9. Notice we've counted from 1 here, which is the carboxylate, down to 9. And again, keep in mind the omega and the deltas will have a bit of differences because we're counting along different sides. See that? We're starting at different points. But the delta here, delta 9, it's always the starting carbon that you define as your, where the double bond is, right? Delta is by far and away the most used. But in very long ones, the omegas start getting used more because these numbers get stupid. Like 20-somethings, 30-somethings, that sort of stuff. And so it just doesn't become meaningful anymore. It's difficult to work with. You can also get things here where like there's some very famous types of fatty acids that humans are the essential fatty acids that have double bonds down here in these last ones. And that's why they're the essential fatty acids, and it takes more energy to digest them and make them. And yeah. So anyways, we'll get a bit more into it, but I don't think I'm going to be able to cover the whole fatty acid was been researched. But it actually is quite something. I'll give you the basis that you can go out in the publications and understand it. It's basically my goal in this class. Okay, so this is the omega carbon atom again. And with the omegas, you actually start counting from the distal end, right? Which is the last one, the methyl group. Notice that is the only methyl group in the entire chain, right? Everything else is methylenes or CHs or carboxylates. Okay? And so again, this is the omega. It looks like a W. This would be the omega 7 double bond here because again, we're counting from the other side. Okay? It still does the one that it starts with. It's just starting from the distal. All right. So the fatty acids in biological systems, these are going to contain an even number of carbon atoms between 14 and 24 typically. 16 and 18 are going to be your most common ones. They're going to have an unbranched hydrocarbon chain in animals. You can actually find branched ones in things like plants and stuff. A saturated or unsaturated with double bonds, unsaturated being with double bonds, and a cis configuration alkyl chain. Short chain length in the unsaturated will enhance the fluidity of your fatty acids and their derivatives. You may already start you may start actually making connections by now. We had talked about fluidity with what? Cell membranes, right? Yeah. So you may start connecting things. Yeah. I know. Okay, so these are some of the naturally occurring fatty acids in animals, and right here we can see the number of carbons. These are the number of double bonds in them. This is the common naming for them. Systematic name is here. I'll be honest, most of these don't actually use the systematic name almost across the board because biochemistry has so many biologists in it. They have a tendency to use the common names. I don't know what it is about biological fields, but they really like their memorizations. So I'm going to be honest, like this is by far and away the more common one. If you're going to see oleic acid or oleate or palmitate, you're going to see them in this name. You basically will never see these unless it's a chemist. Yeah. Which is a shame because these are actually really nice, right? They're really easy to read. Like they tell you exactly what's going This is actually the formula right here. Notice, by the way, the 8s are the actual common name that should get used because you're always in the carboxylate, right, in physiological conditions. The ic acids get used a lot, and that's actually a mistake. But for your information, yes, you'll see the ic acids. Most of the time, probably. Okay? Whatever. I guess you can figure it out, right? Lipids function as actually a lot of things, but the main ones we'll talk about is the fuel molecules, of course, highly concentrated energy stores, signal molecules and messengers in signal transduction pathways, components of membranes, that's all one thing, the essential component of biological membranes as well. The principal lipids in eukaryotic membranes are going to be phospholipids and glycolipids and cholesterol. By the way, is cholesterol the cause of heart disease? No? Yeah, okay, good. You've finally got away from Harvard's grip. Do you know Harvard spent 30-something years and billions of dollars trying to prove the lipid theory, which stated lipids and cholesterol caused heart disease? And the entire research body for that was eliminating over 30 different countries to remain with a set of 2, Australia and the United States, which are the only 2 that had a trend like that. And you can pretty much tell that, like Italian cooking, lots of olive oil, right? Northern European cooking, lots of fatty acids, but they're in very good shape, very low heart disease. Anyways, common attributes, membranes. Membranes are going to be sheet-like structures. They'll be 2 molecules thick. They're going to form closed boundaries. They're going to consist mainly of lipids and proteins with linked carbohydrates. They're going to contain lipids, small molecules with hydrophobic and hydrophilic moieties that will form our lipid bilayers. And again, remember, the lipids are interesting because they have the fatty acid chain, and that is hydrophobic, right? And then we have a head, and that is hydrophilic, okay? Sometimes we'll add something to make it more hydrophilic on there, and sometimes we'll have more than one chain on the rear so that we get the correct angle. They're going to contain proteins embedded in the lipid bilayers. These will have distinct functions, and those are those protein embedded in the cell membrane that we had looked at before with those anchors, okay? They're going to be asymmetric, non-covalent assemblies. They're going to be fluid structures. Please don't think of them as solids. Okay, they're not solids. They're a fluid mosaic. Okay, they're a heterogeneous fluid. They tend to be electrically polarized. You typically have a bit of a charge difference from one side to the other. Okay. These are phospholipids. These are the major class of membrane lipids. So phospholipids are composed of these units. 1 or more fatty acids. These are the fatty acids here. In this case, we have 2. Platform to which the fatty acids are attached. More often than not, glycerol. But you can also have like the sphingosine lipids. We'll also have a phosphate group right there. That's that same phosphate group that you know and love. And then we'll have an alcohol that's attached to the phosphate. Notice this does make this a bit more polar on the end here. This part right here is an acyl linkage. Remember our acyl linkages? Okay, has the carbonyl. Yeah. And these fatty acids here, we have 2 of them, right? What is that going to do to the angle you can make these things at? Anybody know what a one that's not a bilayer is called? So a lipid bilayer cell membrane, right? Okay. Micelle is the one without another layer inside. Okay, so all the fatty acid tails just point next to each other. If you have fatty acids that have very polar heads, they'll assemble into micelles because they can go at a very tight angle, right? Joining together. If I stack my hands, the angle I can do that at is very tight, right? Or if I go the broad way, like a 2-1 would be, that angle's much more constrained, isn't it? Okay, and so you can make a larger size, and it doesn't collapse down to micelles. Cool, huh? Okay, so phosphidate, this is going to be your simplest phosphoglyceride. The phosphoglycerides are going to be phospholipids that are derived from glycerol. I know, the naming is actually fantastic. Phosphoglycerin, or glycerol, glycerin being another name for glycerol, ides. The OH groups, C1 and C2 of the glycerol, are going to be esterified to the carboxyl groups of the two fatty acid chains. And so here we can actually see that we've made esterification or ester linkages like we did in organic chemistry, right? And those are our acyl groups. Again, these are our acyl groups. They're going to have the fatty acid chains here. It is a convention to just label these chains with R groups of some sort and not write out the whole things because they're very long, right? Everybody see glycerin right here, or glycerol? 3-carbon chain with 3 OHs normally. Okay. But we've done ester linkages. Okay, and there's our phosphate right there. Everybody see it okay? Fantastic. Again, this one is the simplest one. This is another name for this, which is the diacylglycerol 3-phosphate. You may actually recognize that from other places in your cell biology and stuff. So our common alcohol moieties of phosphoglycerides, the major phosphoglycerides are going to be derived from phosphidate, which is the one we just looked at, which is the simplest one. Notice that we don't actually care what these chains here are. They're all phosphidates. So we could have a 10, 12, 8, I don't care. Okay? It could have double bonds in it. Again, I don't care, right? We're just looking at the phosphoglycerides and phosphidate. Okay, so an ester bond is going to form between the phosphate group of phosphidate and the hydroxyl group of an alcohol. We can link it to things like serine, ethanolamine, choline, glycerol, N-acetyl. So we can make many derivatives of the phosphidate. And here's the serine-linked one. Again, we've done a phosphodiester bridge, just like we had on like DNA and RNA and stuff, right? Okay. And here's the one with link to choline, linostol, ethanolamine, etc. Does anybody have any questions about that? Come on, this is like, ooh. Oh.
So the ones you showed us before,
Mm-hmm.
Or
These are the heads. These are what that's serine.
Yeah.
And then that's going to be linked through esterification right there. And see the red alcohol?
Yeah.
Yep, that's what's going to link there. Yeah. That's why the amino acid serine is one of the only amino acids you see here. Because you have to have an alcohol, right? Did that make more sense? Yeah, it's still a phosphoglyceride. So it still has the phosphate here. Here's the glycerin.
Oh, that one is, yeah. I was talking about just the regular one, like the one before.
Oh, yeah. These are the heads. Good. This is a selection of things you can link to it.
Yeah, okay, that makes sense.
Oh, no, you're good. Please do. Like, I know that I cannot possibly understand all the things that you guys would have questions for. And so I just have to shoot it out and please question. Okay? The class very much depends on it. Okay, so sphingomyelin, this is going to contain a sphingosine platform. Sphingosine, this is an amino alcohol that will contain a long unsaturated hydrocarbon chain. Sphingomyelin is a common membrane phospholipid. This will have that sphingosine backbone. And this is it right here. So again, this is that sphingosine. And right here, we have the sphingomyelin. Notice the phosphate here linked to this group. Okay. And then we have our acyl linkage, that would be our fatty acid right here. Can everybody see that okay?
So is that R1? Would that be with
The sphingosine is the blue.
that blue?
Yeah. The fatty acid is the green. Yeah, and actually, if you pay really close attention, all the coloring is consistent. Yeah. So, like, instead of glycerin, which was our blue before, we get sphingosine in these cases. Okay? Does that make a bit more sense? Yes, there's a sphingo. Sphingo gets used again. we'll get into some sphingos. Yes, indeed. We are merging together, aren't we? No, you're right. There's a sphingo group. It's this up here. It looks like that. It's a derivation. Yeah. And we'll get into it in a bit, but we gotta get through the phospholipids first. Anyways, the glycolipids, these can include carbohydrate moieties. The glycolipids are going to be lipids that contain a sphingosine backbone with 1+ sugars attached to the primary OH group. And so these are actually what you're seeing right here. The sugar residues are always going to be on the extracellular side of the membrane. Cerebroside is the glycolipid containing a single glucose or galactose residue. So this is a glycolipid. This is the specific glycolipid cerebroside. It is a type of glycolipid. Okay. Again, we have a glucose or galactose here. That's our sugar unit. In the other glycolipids, this would be some sugar unit or some multiple sugar units. Like it could be something like an A, B blood type, that sort of stuff. Okay, it can be quite extensive. Yeah, and this is, we're starting to get into what you're mentioning before, which is the sphingosides binding to different things to anchor them. Okay. Anyways, again, fatty acids, sphingoside. Okay? Or sphingosine, I should say. In this case, not sphingomyelin, though. Okay, so cholesterol. This is actually the steroid that's built from 4 linked hydrocarbon rings. Yes, these are hydrocarbons that have then been rearranged into rings. It'll contain a linked hydrocarbon tail at one end and an OH group at the other end. And so here we see the OH group. Right over here we see the hydrocarbon tail over here. It's oriented parallel to the fatty acid chains of phospholipids in your cell membranes. The OH group is going to interact with phospholipid head groups. And so cholesterol is actually an important building block of your cell membranes. Anybody know why cholesterol get used? Okay, it's a fluid, right? So do you want your cell membranes to always be really fluid and bending and allow things to go punch through them? And no. Not quite strength, more stiffness. Yeah, that's a bit better of a way to... So it makes it a bit stiffer and harder, okay? Less springy, less fluid. And you can kind of see that if you look at this. This has a lot more nonpolar surface area that can't bend as much, right? The other ones can rearrange very easily. So archaeal membranes are going to be built from either lipids with branch ether lipids with branch chains, not either ether, as in organic chemistry ether. Okay, so nonpolar chains are going to be joined to glycerol by ether linkages, more resistant to hydrolysis than ester linkages. So again, these are the ether linkages here. See that? The ester linkages had a double bond carbonyl right there, right? The alkyl chains are actually going to be branch. They'll be saturated hydrocarbon repeats. They're more resistant to oxidation than the on-branch chains. So again, here we have the branch chains right there. The stereochemistry of the central glycerol is actually going to be inverted. So you may have noticed that our stereochemistry of our glycerols before were like so. We have flipped it on this one. See that? Instead of the bottom, it's the top. Okay. And so we've flipped our stereochemistry the other way. You know what? Off the top of my head, I don't remember. Maybe I put something in my notes, but I kind of doubt it. I'm not sure if there is a why answer. That would be a great thing if you want to tell me why. I can put it in the slide. And if they figured that out at all. Yeah. But yeah, I don't remember if they actually ever did anything. Like the rest of this is all to prevent oxidation because they're archaeal. They tend to live in extreme environments, right?
So the non-polar chains have linkages, but the polar have an S, or S, right? And the S
Yeah, well, this has an ester, phosphodiester, but everything else doesn't. Yeah, there's no esters in here except for the phosphodiester.
yeah, but I'm just talking about like in the, if you go back, like for the phosphate.
Oh, this is the difference between prokaryote, eukaryote, which was what we were doing before. These are the ester linkages. And then these ones are archaeal membranes.
Oh, okay.
Yeah, so a different evolution pathway. And very specific difference in the evolution pathways. Because of the extreme conditions, high oxidizing conditions typically, they need to be very resistant in their cell membranes to oxidations. Whereas in us or most prokaryotes, we are going to control our environment and we won't allow ourselves to be in such oxidizing conditions, right? Or we won't survive them. You can think of it that way too. And so we're actually more concerned with being able to recycle these easier, okay? And make new cell membranes really fast. Does that make sense? This would actually make it much more difficult to recycle. This will take a lot more energy to take these and break these down, okay? Whereas, remember our organic chemistry? Making those esterification linkages is actually very easy, right? Okay, again, this is why, one of the big reasons why these can live in extremely harsh environments, very harsh habitats, right? Like so. Did that make sense? Did we cover everything there? I know there's a cluster of questions. I think we got it. But yeah, if you actually tell me why that is, that'd be interesting. I've never seen it. But, you know, this literature doubles every 42 days. There's a lot I haven't seen. There's a lot everybody hasn't seen. Oh, man. So membrane lipid is an amphipathic molecule that's containing a hydrophilic and hydrophobic moiety. This is what makes it have the entire ability to make a membrane in the first place. Okay? Amphipathic or amphiphilic, another way of saying that same thing, molecules. This can be a molecule that contains both a hydrophobic and hydrophilic moiety, i.e. it has one side that's hydrophobic, one side that's hydrophilic, right? And so they can interact with water on one side. They'll self-arrange so that the hydrophobic ends always interact with each other. So membrane lipids are going to be amphipathic molecules. They have to be because one side's going to touch water, right? The other side needs to not want to be touching water. So that stays together. So the hydrophobic moiety is the fatty acid tails. The hydrophilic moiety is the phosphorylcholine. You'll notice, by the way, a lot of these ones that use in cell membranes actually have groups on the end, the hydrophilic end, that would make them more hydrophilic than a normal fatty acid. This right here is the hydrophilic unit. That's the polar head groups. It's represented typically by a circle of some sort, like this. The hydrocarbon tails are typically represented by some sort of straight or wavy line, like so. Okay, you just sort of make a line. And bend it as you will. Notice that some of these this has on saturation. See that? There's a double bond in that one. And so that would have a kink in it. You can actually see the kink down here. Okay. Can everybody see that you can actually kind of see how these would work in a cell membrane, wouldn't they? So they kind of scissor each other like that. Wow, is that an okay terminology term to use? Okay. And then these are the heads right here. This is the archaeolipid. Notice that has those branches coming off of here. There we see, okay? That we just talked about. And this is the sphingomyelin right here. Notice that one of these is the sphingosine. That's the phosphoglyceride. So the membrane formation is going to be a consequence of the amphipathic nature of the molecules. This is actually a mycel. This would be a globular structure with a polar head groups on the outside surface and a hydrocarbon tails sequestered inside. Has anybody ate way too much fats and their pee looks like cloudy? It has like refraction points, little bubbles. No? Man, you guys need to pay attention to your lives. That's actually kind of a bad sign. If you start seeing that consistently, you need to get to the doctor, okay? But if you just like ate a whole pack of beef jerky or some stupid thing, it's fine. You can have that happen every now and then. Anyways, those are micelles, okay? This is actually how soap works too. Does everybody know how soap works? Okay, so in soap you have fatty acids that have like a sodium or potassium on the head here, and it makes it more soluble right here. And so they arrange like this. Grease gets trapped right here, and it gets solubilized in micelles that then come off into the water, right? And get washed away. You can do the same thing to your cells, by the way. That's why you can use detergents and soaps to destroy cells and make them come apart, like we saw in chapter 4. Anyways, lipid bilayers. This is what we're usually talking about when we're talking about cell membranes. So here's our lipid bilayer. This is going to be a bimolecular sheet. You're going to have two lipid sheets right here. You'll have, again, the hydrophobic tails. They're going to point on each sheet, interacting with one another. This is going to be what forms your permeability barrier. So remember, both sides will be a water, right? Some sort of water solution. And so this right here is what creates that semi-permeable membrane. The hydrophilic head groups will interact with the aqueous medium. Your hydrophobic tails point into each other and create this separation. Anybody have any questions about that so far? Okay, fantastic. One thing you have to keep in mind that I know it looks like a solid wall here when you look at these. It is not a solid wall. Okay? Like CO2 and things that are neutral can get through this pretty easy. Okay? And you can have exchange of water even through this because it's still a neutral molecule, right? Yeah. It's charged things that have a really hard time getting through or fairly large things. I mean, not that large, but you get the idea. That's what makes that the semi-permeable membrane. All right, so the lipid bilayer formation will be spontaneous. They would self-assemble if you put lipids together. You'd form micelle. If you put phospholipids and glycolipids together, they'll form a lipid bilayer. The phospholipids and glycolipids, because of your space taken up by two tails, do not form the small micelles. In fact, they cannot assemble into small micelles typically, the way the single-tailed salts of your fatty acids would, like soap would. The phospholipids and glycolipids are going to spontaneously form your lipid bilayers in aqueous solutions. This is stabilized by several factors. We have our hydrophobic interactions. Another way of saying hydrophobic interactions is hydrophilic exclusion interactions, right? The van der Waals interactions between the hydrocarbon tails. Remember, these are pretty long hydrocarbon tails, so they actually have fairly strong van der Waals interactions, right? The electrostatic and hydrogen bonding attractions between your polar head groups and the water molecules. The lipid bilayers will have an inherent tendency to be very extensive. Lipid bilayers will tend to close on themselves so that no edges will be exposed to the hydrocarbon chains to forming to the water, right? They don't want any of those edges exposed, so they're never like this. They always close back in on themselves. And they always make a sphere then of some sort. It can be a lumpy sphere. Sphere. All right. These are going to form compartments if they need to. The lipid bilayers are self-sealing, So if you make a hole in your lipid bilayer, it's going to be energetically unfavorable, and it will be filled in. It will either contract to fill it in, or it will bring in more material. These are lipid vesicles, otherwise known as liposomes. You guys remember liposomes, right? Fantastic. Okay, so again, we just have a lipid bilayer. It's a smaller one, so we can call liposome, excuse me. They're going to be aqueous compartments. They're enclosed by our lipid bilayer. They're used to typically study membrane permeability or to deliver chemicals to the cell, or to some degree, versions of these are used in our body, of course, to send off proteins and such to our cell membranes. Anyways, we have an outer aqueous compartment. We have an inner aqueous compartment. This is the lipid bilayer right here. All right, so liposomes, these containing trapped ions or molecules, can be synthesized. They're formed by suspending in lipid in an aqueous medium. You then sonicate, which will agitate, which is, have you guys ever heard of sonication before? No? Okay. So what that is you take a bath, and on the side, you essentially have really high-frequency speakers, okay? So high-frequency you can't hear them. And what this is going to do is it's going to vibrate the insides right here. This actually causes like bubbles of air to form and then collapse, and causes hypersonic agitations, okay? Anyways, it's really good for breaking stuff up, including like breaking up stains on glassware. Okay? But anyways, again, agitation by high-frequency sound waves. The ions or molecules will then be trapped. So we have glycerin in here and H2O. This is a phospholipid. What ends up happening is you do sonification. This is going to cause this to break up and get intermixed in there. Okay? And it basically breaks up your structures kind of unevenly. And so you end up with the phospholipids coming up into the water. What's going to happen then is they're going to form spontaneously into our wonderful liposomes. And those liposomes will then entrap some of the solution that has the glycerin in it, right? Okay. And again, these will be trapped into those compartments. The specific membrane proteins can then be embedded by solubilizing proteins in the presence of detergents, and then you can add them to the phospholipids. So for example, if you wanted to, you could then do a gel filtration, separate this out. You could then go and embed specific membrane proteins. So these could just literally go onto cells, incorporate the proteins you want, have the substrates you want, and bam. Okay? And then they get introduced to the cell. You can also do other things like deliver drugs to it, deliver specific proteins or enzymes that you want to deliver, etc. Okay? It's quite versatile. And it's fairly easy to do. Yeah. Surprisingly so. Do they do a lab like this at all? Downstairs? It's actually kind of a shame. Because this is like cheap and easy to do. Yeah. So most properties of planar bilayer membranes can be studied experimentally. They'll spontaneously form across a hole in a partition between aqueous compartments by spreading a membrane-forming solution across that hole. So right here we have a partition, there's a hole in it, and if you were to go ahead and put in membrane, your components of your membrane, your phospholipids or whatever, it will actually form a membrane and split up these two compartments for you. The electrical conduction properties of your bilayer, these can be studied using electrodes. So if you have two electrodes, then you can actually study what different electrical conditions, what different ionic conditions can do to these. Okay? And you can actually get electrode conditions and electrical conditions, ionic conditions, that will cause the breakup of these lipid bilayer membranes, or them to stiffen or change property. So it's actually quite interesting. Like you can pulse certain electrical signals, which would actually cause it to stiffen really a bunch, become very hard. Yeah. Anyways, lipid bilayers, these are highly impermeable to ions in most polar molecules. The lipid bilayer membranes are going to have a very low permeability for ions, and again, most polar molecules. Your permeability of small molecules will be correlated with their solubility in a nonpolar solvent relative to their solubility in water. This is typically referred to as the blood-brain barrier, but the blood-brain barrier is actually a much more specific barrier of this same condition that exists in the blood going to the brain, right? But this is what causes the blood-brain barrier, okay? The water is going to be an exception due to its low molecular weight, high concentration, and the lack of a complete charge. And so you do end up squeezing water through, but part of what causes the water to squeeze through is not just that small and it has no charge, it does have a partial charge, right? And so it's not exactly the most favorable thing to squeeze through. But you have to remember these things are surrounded by water solutions, right? So they're getting hit all the time by water solutions. And so it happens. This is actually the permeability coefficients, which is P for permeability, of ions and molecules in a lipid bilayer. And so right here we can see these are increasingly permeable as we go up. And here we see fairly charged ions here. And notice they have very low permeability, right? And so they're not going to get through that lipid bilayer very well. Here we have things like glucose, urea, glycerol. Again, these are fairly polar molecules, not super small, right? And again, these are pretty low permeability. And then indole and water. Anybody have any questions about that? No? Okay, so proteins carry out most of your membrane processes. The membrane proteins will allow your transport of molecules and information across your membrane. You're all probably pretty familiar with this. This tends to be what biology kind of concentrates on when they're going to concentrate on something with the membrane. The membranes vary in protein content. This can range from less than 20% to as much as 75% protein, which is quite something, right? What a range. Membrane proteins can be visualized by your SDS, polyacrylamide gel electrophoresis. The types of membrane proteins in a cell are going to be a reflection of your biochemistry that occurs in the cell. If you need glucose across it, guess what you're going to have for a membrane? Something to get glucose across it, right? If you need a lot of potassium being released and brought in, guess what you're going to have? A membrane that facilitates that, right? Membrane-bound protein that facilitates that. If you have to exchange sodium and potassium repeatedly because you're a nerve cell, guess what's going to happen? You're going to have proteins to do these, right? Okay. So the lipid-rich membranes of myelin are actually going to form an electrical insulator around some nerve fibers. Myelin is going to be a membrane that serves as an electrical insulator around certain nerve fibers. I absolutely hate this terminology. It is fucking ridiculous. Who here, nobody thinks, hopefully, that you have electrical signals going through you like wires, right? Like little zaps of electricity. That's not how that works. It's a polarization change that propagates along the nerve. Okay? So calling it an insulator is just not right. It's just, frankly, a wrong terminology used completely, utterly, and it's in every biology book ever. Okay? But whatever. It insulates them from the polarization changes. What it has is it has a low content of protein. It'll play a role in the rapid transmission of your nerve impulses. It's formed by Schwann cells wrapped around the axon. Again, that's the part of the neuron that conducts your nerve pulses. This is the axon right here. It's literally little channel connections that are going to release and then cause another release later. And we'll get more into the axons and nerve impulses later, but here's your overall structure. Can everybody see it okay? Okay, here's a Schwann cell actually engulfing right now. And then we had multiple Schwann cells begin to grow around. Well, we have the Schwann cell growing around and wrapping, and it builds up and up and up and up, and that gives us our myelin. Okay. Does anybody have any questions about that so far? Fantastic. That's what it looks like, by the way, if you were to look at a dissection under a microscope. The protein composition of plasma membranes can vary with cell type. So your protein, your membrane proteins actually, again, can be visualized by the SDS polyacrylamide gel electrophoresis that we talked about in chapter 4. Your membrane's going to perform different functions containing different repertoires of proteins. And again, we can tell the cell type where they match. We can then take these proteins, we can figure out what they are, and build up our knowledge slowly but surely, right? Okay, so integral membrane proteins. Integral membrane proteins are going to interact extensively with the hydrocarbon chains of membrane lipids. These will be released by agents that compete for these nonpolar interactions. Most will span the lipid bilayer. Again, integral meaning within, right? Okay. Or integrated, integral, same root. Okay. Peripheral membrane proteins. What are these going to be on? Outside, yeah. Well, on the periphery, the outer portions of the lipid bilayer, right? Yeah. Not the outside cell necessarily. So peripheral membrane proteins can be bound to membranes primarily by electrostatic and hydrogen bond interactions with the head groups of your lipids. These are going to be disrupted by adding salts or by changing pH, because again, that would change that hydrogen bonding characters, those interactions. They're going to often bound to the surfaces of integral proteins. So you can have them bound to the integral protein, which would then serve as its anchor, right? To keep it in the attached. May be anchored to the lipid bilayer by a covalently attached hydrophobic chain. And that's kind of what we would see. We saw hints of that in like the sphingosine, right? When we were looking at that. These right here, this is the overall diagram here. Okay. And so, again, this is a integral. This one is integral, exactly. This one is peripheral. Peripheral? Still peripheral. Yeah. This would be something like a sphingosine. Mm-hmm. Got it. Yeah, it just acts as a little anchor. Okay. So the proteins are going to interact with membranes in a variety of ways. Your membrane spanning alpha helices are going to be one of the most common structural motifs in your membrane proteins. It'll literally be a alpha helix or usually several alpha helix to make a rod. It'll have hydrophobic groups around it. And so then it can go through your cell membrane and you can actually have a multitude of alpha helix making a channel. Okay? Bacterio orthodicin, excuse me. This is the light-powered proton pump in archaea. Integral membrane, I so rarely get to say this word, which is, you so rarely get to talk about archaea, right? Yeah, because we just don't deal with them a lot. Anyways, really cool. Again, it's the light-powered proton pump. In archaea, it's an integral membrane protein. Actually, this function pretty much tells you it has to be an integral membrane protein because it has to, transfer it through. It's composed almost entirely of alpha helices. And so this is what this actually looks like. See these little rod things? Those are what? Alpha helices. Yeah. So basically, all this is composed of is alpha helices with little turns to connect them. Okay? And it does have a little bit of beta sheet here. But yeah, it's almost all alpha helice. And see how they are basically used like little rods, like logs, to make a structure? Okay. And so it gives literally a channel that you can pass hydrophilic stuff through, right? Or in this case, a proton. Okay? Cool, huh? And this is looking down into it. You can see the channel Come on. That's just beautiful.
So they make like like the circle around it.
Exactly. Yep. So yeah, this is the little circle around here. The channel is going to be right here. Keep in mind, it is going to be light powered, so it brings them through. So this thing's going to change. But this right here is the cytoplasm, in case you're wondering. The beta sheet's on the outside. The membrane spanning helices of your Bacteriorhodopsin is containing predominantly nonpolar amino acids, which is probably not a surprise to you because it has to interact with that nonpolar region, right? Yeah. The nonpolar residues are going to be in contact with your hydrocarbon core of your membrane or with one another to keep them bound together in that structure. Remember that everything on the outside of this assembly is going to end up being hydrophobic. That's what's passing through the tunnel, right? So the polar and charged residues will tend to be found in the cytoplasmic and extracellular regions. This is what allows it to interact with the cytoplasmic and the extracellular hydrophilic regions. This is porin, by the way. Again, we can form channel proteins with beta strands. That also is a way to go. Basically, you can form them with anything that forms structures, right? The porin, this is going to be the protein that will form the outer membranes of bacteria. They're formed from a single antiparallel beta sheet that's then curled up to form a pore channel. This is the pore channel. You can see down through it. This right here, you can see the beta sheets and they're anti-parallel. See that? They go the opposite directions each time. Okay. The outer surface is going to be nonpolar. The inner surface is going to be hydrophilic. This will be filled with water. It usually makes pretty big channels when you see them. The porin is going to contain alternating hydrophobic and hydrophilic amino acids. They're going to be along each beta strand, and this is what we see right here. See how they alternate between them? And it's literally the anti-parallel, remember, causes you to have pointing in the opposite direction R groups. Remember that? And that's what you're seeing, is you're seeing that opposite directions for the anti-parallel. Okay? Okay, so prostaglandin H2 synthase 1. This is an endoplasmic reticulum membrane-bound enzyme that's going to promote inflammation and modulates gastric acid secretion. It's a homodimer. It's going to lie along the outer surface of your membrane. Though do keep in mind this is the endoplasmic reticulum we're talking about, not the outside of the cell. Okay. This is going to be bound by a set of alpha helices. This is going to extend from the bottom of the protein into your membrane, and that's what it looks like. So again, it's going to be classified as an integral membrane protein. This is because detergent is actually required to release the protein. And so this is one of those cases where the definitions can become somewhat difficult, right? You may be tempted to categorize this as peripheral, but this is not two different proteins. This is one protein altogether. And so this one does have an integral portion to it, which makes the protein itself integral. Okay? And the big test usually is if you can get these to detach from just putting salts and stuff in there, then that's peripheral. Okay? If these things take detergent to detach, they're usually integral. Okay? That's usually the test that separates these things. Anyways, this is the hydrophobic amino acid side chains that make up that anchor. Everybody can see that nicely? Okay.
So it's classified as an integral, but is it needed? It's not using it as like a channel or anything, right?
No. This is just an anchor. It's just permanently anchored in. It's not meant to be released. Yeah.
Like a porin is like a channel.
It is. The porin is a channel, and that one is integral as well.
Is it like in the
Okay, this one, we're still talking this one right here,
No,
or the port.
like the port is like, I understand that one how it's on the outer because it's like used, it's not used as a channel. Like,
The port? The porn's used as a
no,
channel.
I know the port is.
Okay.
So it's like.
Okay, we're talking about this one then.
Yeah, that one's not a channel. It is on the
No.
outside, right?
Yes, it's on the outer part of the endoplasmic reticulum.
Yes, on the inner part. But
Yeah.
then the pores, it's also, it says it's on the outer of the membrane of bacteria, but then how is it?
Well, there's several different membranes in a bacteria. This is the outermost cell membrane. And yes, it extends all the way through the membrane. Yeah. And this is a pretty good figure of that. So you can see there's parts of the protein on both sides. Does that make more sense?
But is it like integrated in the membrane, like so that it can go all the way through?
Yes. Yeah. This porin is. Yeah. So that outer cell membrane, the one that's on the outside part of the cell that prevents things from getting through, okay, this one is going to be integrated throughout that entire membrane. And so you have direct channel all the way through. Yeah. And all channel proteins have to do that.
So do the hydrophilic heads in the cells, do they kind of like wrap around it?
The hydrophobic, oh,
Like, do they kind of like,
yeah, if you were to look on this, they're right over here.
okay, yes, okay, that's what
Yeah. Yeah. They should not be in here.
No, I know they should be in there. But I was thinking kind of like if the membrane, the membrane goes like here, and I was thinking the channel was on here, and I was like, how does it get in here?
Oh,
Integrated in.
yes. This is the cell membrane right here. It's wrapping around like
Yeah,
that.
it wraps around.
Okay.
I was just like,
Good. Okay. By the way, an interesting side note to this is you guys may not have made this connection yet, but like you need the hydrophobic R side chains pointing outwards towards the hydrophobic portion of the cell membrane. But the hydrophilic ones going inwards are actually just as important. Because if you left this with no hydrophilic ends, enough space in here, remember cell membranes and lipid bilayers will plug small holes on their own. Okay? It would just plug it. Right? Like people don't really make that connection usually, but both parts are just as necessary. You need both. Okay. Did we have any questions on this one? Okay, fantastic.
sorry.
Oh, no.
Like when I think, well, I get to look at it, it just takes my brain a minute to process it, and then I have questions. We're a few slides ahead, so I hate asking you to go back.
That's fine. Okay, so this is arachidonic acid. It's a hydrophobic molecule. It's going to be generated by the hydrolysis of membrane lipids. And again, that's the arachidonate right there. This is actually the oxidation here. This gives you prostaglandin G2. Prostaglandin. We heard about this before? Anybody notice anything that's like a shape that looks like it might join into some sort of ring or something? Okay, we'll make those connections later. We'll have figures. Okay, so this is going to create the prostaglandin G2. Peroxidase right here is going to further oxidize this, and that's going to give you the prostaglandin H2. The arachidonic acid is going to reach the prostaglandin H2 synthase 1 active site. This is going to happen through a hydrophobic channel. This right here is your hydrophobic channel. Right there. Everybody see that okay? Fantastic. Okay, so the aspirin actually inhibits the cyclooxygenase activity of prostaglandin H2. Again, cyclooxygenase right there. So the aspirin will inhibit that cyclooxygenase activity. It's going to do that by transferring its acyl group to the serine 530. That's that little red thing there. See the serine 530? Okay. The serine 530 lies along that path to the active site, and this will block that channel. Again, this is the active site right there. See the little molecule in there? Okay. And so that would actually then covalently modify the active site, block it, and now we can no longer do the reaction, right? And that is one of the inflammation blocking activities of aspirin. Cool, huh? All right, so some proteins are going to be associated with membranes through covalently attached hydrophobic groups. The soluble proteins, these can be associated with membranes if hydrophobic groups are attached, like palmitoyl group, which is attached cis by thioester bond, farnesyl group, which is attached cis at the carboxyl terminus, the glycophosphatidyllinositol, GPL, GPL being the much easier one. This is a glycolipid again. This is the anchor that will be attached to the carboxyl terminus. These are actually what those look like right there. And so here we can see right there the attachments. Notice again, that's that thioester bond right there. Okay. Which amino acid has the thiol, by the way? Cis, right? Which is why we're attaching it cis for each of those. Because it's thioester bond. Thiol bond. Okay. And this is what it looks like for the GPI anchor, by the way, right there. Yes, that's that same sugar pattern that we looked at before. Okay. Does anybody have any questions about that? Okay. Notice that's the carboxyl terminus right there of our protein amino acid, okay? Up here. This is the lipid anchor right there. And this is just the structure that connects the two. Okay, so the free energy changes for the transfer of individual residues from hydrophobic to an aqueous environment are given in this table right here. These can actually be used to estimate the free energy change when a helical segment would be transferred from the membrane interior to water. And again, this would give you that polarity scale, right? How likely it would be to attach to the hydrophobic versus the aqueous environments. Okay? What do you notice about these? Yeah, well, things that are charged, very hydrogen bonding, prolific, they're going to give off energy from going from the non-polar environment to the aqueous, right? And then up here, we have things that would, they would take energy to force into the aqueous environment, wouldn't they? Okay? This tells you how easily it would be to remove or put in, how well it would bind to that cell membrane. Another way to think about it. The transmembrane helices, these can be accurately predicted from amino acid sequences because the amino acid sequence you're going to have are going to have a lot of R groups exposed to the hydrophobic conditions. That would be these ones right up here, right? If you have any of these ones, what's going to happen? Not going to stay in that lipid membrane, is it? No, it's going to push itself out because it's very energetically favorable to do so, isn't it? Okay. So an alpha helix that consists of 20 residues can traverse lipid bilayers. The free energy change when a hypothetical alpha helix is transferred from a hydrophobic to a hydrophilic environment. Again, this can be calculated for each stretch of 20 residues based on this table right here. Literally, you would go down and just add these together, right? If it's a positive value, it's going to want to be in the lipid bilayer membrane, right? And then if it's a close to zero value, what does that tell you? Doesn't really favor either, does it? Yeah, so it can pretty much just move wherever. And then if it's a highly negative value, what do you get? Not going to go through the layer, right? This is going to stay aqueous. Another way of thinking that, yes, it would very easily come out, but another way of thinking of that is it's very energetically unfavorable to try going into that bilayer. Okay? And so to do that, you would actually have to pump in a bunch of energy. Yeah. Yeah. Essentially, it would try to go in, it collide, and the energy would start eating away at its energy, kinetic energy, right? And then it would be favorable to then repel. Or another way of saying that is it'd be favorable to start making hydrogen bonds. So the water will pull it out. Does that make sense? Okay. The window is a span of 20 residues chosen for this calculation. You can actually shift your window down your different proteins to see if there's any portions that would anchor themselves into a cell membrane or similar structure. Hydropathy index, this is the free energy change for each window. And that's what we were referring to before. That's the hydropathy index. That would be a large positive, 0, large negative sort of number. And that would tell us whether it would go into a cell membrane or not. Again, the hydropathy plot is the hydropathy index plotted against the first amino acid at the window. And this is right here. Notice this right here again. This is the criterion level, a large positive value, right? And so this would be where we would start seeing these anchor themselves into cell membranes and stay there pretty consistently. Okay? And so this right here is actually a single alpha helix that's in the glycoporin that we just talked about before. Okay? So the glycoporin here, that's one of those alpha helixes. So the peak at plus 84 kilojoules per mole or more and will indicate a possible membrane spanning alpha helix. This does not prove a segment is a transmembrane helix. It just proves it could be. Okay? It could also just be hydrophobic because it's going to do a hydrophobic interaction with another part of the protein, right? Or it's going to hold together subunits. It could be not exposed in the right way to be able to get into the bilayer. This is actually what that looks like in a very sort of open chain sort of way, so you can see what's going on, rather than it being, wrapped up and difficult to see. Everybody understand what's going on there? Okay. So here we have a bunch of hydrophobic ones on that alpha helix, and so that's the integral portion of this. Okay. Again, this is the no strong peaks observed right here. So we did the hydropathy plot for porin, and we see there's no particularly strong peaks there observed. The biological membranes are not rigid, static structures. You have lateral diffusion. This is the process by which lipids and many membrane proteins are going to be constantly in lateral motion. So they're constantly moving throughout that side of the membrane. This can be visualized for proteins by a technique of fluorescence recovery after photobleaching, or FRAP, FRAP, like Frappuccino. The cell surface component is going to be labeled with a fluorescent chromophore, and it'll visualized via fluorescence microscopy. The fluorescent molecules in a region will be destroyed, i.e. bleached. That's taking away the color, right? Bleaching, removing the fluorescence color, destroying them, by an intense light pulse from a laser. Literally, so we put these fluorescent probes throughout the cell membrane, we bleach a part, and then what should happen if there's no lateral movement? It wouldn't fill back in, right? And the lateral movement, how fast it fills in, shows us that amount of lateral movement, doesn't it? Okay. So the fluorescence is going to subsequently monitors as a function of time. Again, it's going to use a low light level. This is going to prevent your further bleaching because remember that the reason we had those plates in that little baggies and all that, and we were so careful not to keep them out of the box and all that, is that those would bleach over time. Anything that is a dye of any sort will fade over time with exposure to light. Okay? That's why your clothes always fade even if you don't wash them. You just keep them under light. Frap recovery. The recovery equals a process resulting in an increase in the fluorescence intensity. It's going to occur if the labeled component is mobile because the bleached molecules will leave and unbleached molecules will enter. The rate of recovery is going to depend on the lateral mobility of the fluorescence labeled component. Average diffusion, average distance, S, traversed in time, T, depends on the diffusion coefficient, which is D. This would be expressed by this equation here, which would be the distance equals 4 times the D, the diffusion coefficient, times the time, and this is to the 1/2 power. This right here would be an overall diagram that shows you what's going on here. Okay, so again, here we have a cell membrane. We have here fluorescence labeling. We bleach one portion of it, and what's going to happen is this is going to move in here, and it will slowly recover. And how fast it recovers, again, shows us how much movement there is, right? Okay. Everybody's good with that? Okay, so our fluid mosaic model, this is going to allow lateral movement, but not rotation through the membrane. So fluid mosaic model will describe the biological membrane organization as a two-dimensional solutions of oriented lipids and globular proteins. The lipid bilayer is both a solvent and a permeability barrier. The lipids are going to rapidly diffuse laterally in membranes. Your transverse diffusion, which is the flip-flopping between the two layers, is very slow, okay? That is far, far slower than the lateral movement, right? Because remember, you have to overcome the hydrophobic and hydrophilic energies to do that, right? Okay. This is what that looks like. So the flip-flop of a protein molecule has not actually been observed. This will preserve the protein membrane, the asymmetry among the membrane of the proteins. This is why like an externally facing protein stays externally facing, okay? Because to do this flip-flopping, you actually have to overcome all that hydrophilic and hydrophobic interactions, break all of them and flip-flop, right? It provides a very large barrier, okay? However, you can actually see this a little bit with the lipids, but again, it's very slow compared to lateral. Does anybody have any questions about that? Okay. Okay, everybody understands the flip-flopping or transverse diffusion versus lateral diffusion? You remember how we did diffusions back in mostly Gen Chem 2, but a little bit organic stuff? This is still that, okay? This formula here is actually the same formula I just derived. So that's simpler to use for this. That's all it is. There really isn't any difference. The same diffusions we talked about when we were talking about like releasing substrates and stuff. How fast an enzyme could possibly go. Similar diffusions. So your membrane fluidity is going to be controlled by fatty acid composition and cholesterol content. Many membrane processes will depend on the fluidity of the membrane. This depends on the properties of your fatty acid chains. The transition from rigid to fluid state takes place above the melting temperature for any given membrane system. And so this would be fluid-like, this red part, this would be solid-like. Ideally, we would be somewhere in the middle grounds. We don't really want to be either, do we? Yeah. We want to be a little bit of both. Fluid-like would come apart very easily, allow stuff through very easily, and would not have a lot of structure or anything, separation, anything. Solid-like, it would be very rigid. I mean, this is a 2-molecule layer of fat, right? A thin layer of fat, how tough is that? No, yeah. Like any touching of it at all by anything, pop, right? So, yeah, being solid-like is not exactly a great idea either, okay? You need it to Have you ever heard the difference between hardness and strength? Did you guys talk about that, Gen Chem, or do you remember it? Hardness and strength. So steel is very strong and nylon is very strong. Yeah, so when you have something very strong, one of the major reasons strong things are strong is because when you put energy into them, try to break them, they bend and can incorporate that energy, right? When you have something very hard, it oftentimes doesn't have that property. And so what happens is, if you were to take steel, you can hammer on it all day and you're going to break it, right? But you can take a diamond and hit one hit with a hammer on a concrete floor on a diamond, it will crack, it'll break. Okay? You can actually drop them and they'll break. You have some weight on them. Anyways, like the big diamonds, they've actually broken them by accident before. Anyways, so that's that difference here. Like being hard does not make it stronger. Okay? This actually gives it strength by being more fluid-like here. Okay, what temperature do you think we normally want to be in this area right here, by the way? Like a physiological temperature-ish, right? Yeah. Yeah. We obviously wouldn't want to be either of these, right? So the melting temperature of phosphatidylcholine containing different pairs of identical fatty acid chains is represented down here. And we can test sort of what would happen if we have these different cases. So as you probably figured out, if we have a longer chain, what do you think is going to happen to that TM? Yeah, it's going to be harder to melt, right? Yeah. And so higher TM. If we have a smaller chain, it's going to be lower TM, right? Incorporating cholesterol is going to raise this TM, okay? The number of double bonds. The double bonds, if we look at 18 here versus 18 there, what happened to the TM? A lot, right? And that's not super surprising because the double bonds kind of make it kink, don't it? Yeah. And that's not going to fit really well together, right? So you won't have a lot of overlap, and your diffusion forces will be fairly low. So these straight hydrocarbon chains of saturated fatty acid residues will interact favorably with one another. This will favor a more rigid state. A cis double bond will produce a bend in your hydrocarbon chain. This will interfere with your highly ordered packing of your fatty acid chains. This will interfere with highly ordered packing of fatty acid chains and will lower the TM. And this is kind of an interesting thing. This is actually where heart disease starts really coming in here, and you can sort of start seeing the tail ends of it. So a lot of heart disease tends to be things where like you're getting micro damage, and then you're trying to repair the damage in your blood vessels, right? And to do so, you have to bring in cholesterol. And unfortunately, when you bring in too much cholesterol, it doesn't always work out, right? What happens when you increase the concentration of a non-polar substance in a polar environment? Has a tendency to start sticking, doesn't it? Shit. Heart disease. Okay. So the long hydrocarbon chains are going to interact more strongly than the short ones. Each additional CH2 group is going to contribute about -2 kilojoule per mole to the free energy of interactions of two adjacent hydrocarbon chains. Remember, negative means more spontaneous, right? Meaning a stronger interaction. This is, again, that packing we can see for the fatty acids, and this one has a cis double bond incorporated. And you can see it does not pack well with the others. It disrupts that. So this right here is going to be with the cholesterol. So the bulky steroid nucleus of the cholesterol is going to disrupt the regular interactions between fatty acid chains. This is going to help to maintain the membrane fluidity in membranes in animals. And so we can use cholesterol as sort of our, you know where you get tables that don't quite stack together well? Okay. You get like a table and it has, oh, this one does. And so how do you fix that? Yeah. That's what we're doing here, aren't we? We're literally putting little wedges in, so they can stack better, right? And so you can, or so you can disrupt the structure. And so cholesterol is kind of like our modulation mechanism that we use along with the chain links, which the chain links we don't always have a choice on, okay? So cholesterol will help that process of modulating our cell membranes to have the correct TM, the correct properties. Does it always work? No. Do we sometimes fuck up terribly? Yes. The cholesterol, this can form complexes with lipids that contain the sphingosine backbone and with lipid-anchored membrane proteins. Lipid rafts. You knew it was coming, didn't you? When I started talking about fluids and floating around on them. Lipid rafts, these are going to be membrane domains containing such complexes. These will exhibit reduced fluidity in that domain. And essentially, they're portions of this that tend to float along with each other. Okay? You can think of a bunch of rafts on a river. And so individually, the rafts don't come apart very much, but they may shift a bit, right? And then they move around each other quite a bit, kind of like continents do. Okay? They may induce conformational changes in membrane proteins to regulating their functional activities. They may function in signal transduction by providing favorable environments for specific protein-protein interactions. Right here, we actually are seeing a lipid raft. This is actually a real lipid raft right here, by the way. See a little pointing and highlight. Okay. So, again, this is going to be cholesterol and sphingolipid-rich membrane domains. And see, we have a grouping of different proteins that need to be near each other, and we want to keep together. And so we make a raft around it, and now that's going to move mostly together. Okay? All right. And we can see it right there. Anybody have any questions there? Fantastic. Okay, so all biological membranes are going to be asymmetric. The outer and inner surfaces for all biological membranes have different components, different enzymatic activities. Probably the best example of this, and the most monitored one, would be sodium and potassium pump in the plasma membrane, which has a directionality to it. Everybody see that? It's very clearly different from one side to the other, right? And so the extracellular environment right here, this is the intracellular environment. This intracellular environment is where the ATPase is. That's going to be allowing the energy to be provided for this process, which makes sense because having ATP trying to come on the outer side of the cell would be problematic, right? Okay, does anybody have any questions there? Okay. So the prokaryotes and eukaryotes are going to differ in their usage of our biological membranes. The bacteria can be classified based on their plasma membranes. So we can classify them as gram-positive. This is having a single membrane that's surrounded by a thick cell wall. Or gram-negative, which has two membranes separated by a periplasm. The periplasm will contain the cell wall. Okay? And that's that gram-negative, gram-positive staining. Too. Okay. You guys okay? All right, so this is gram-positive, by the way. This would be gram-negative. So in gram-positive, your cell wall is going to surround your cell membrane, and it's going to be directly exposed. So when you die, it's going to absorb that dye and bind to it really well, and you'll be able to see it quite nicely. You then wash off any excess, and you get your gram-positive, right? Gram-negative, on the other hand, has an outer cell membrane and a porin. This right here is your lipoprotein. This right here is your cell wall. All together, this entire region is called the periplasm. Okay? Have you guys went through that yet? No, haven't seen any of this yet? Okay. Man, they make you use the gram-negative, gram-positive. They don't tell you anything about it, huh? Those biology people. Silly. Okay. So anyways, the periplasm here is going to somewhat protect this cell wall. It's also going to have a bit less cell wall thickness usually. And so the gram-negative means that it only stains very little, right? You still get to some staining. Okay, so the bacterial membranes, these can be distinguished with your gram staining. Again, the gram staining is going to be the technique for classifying your bacteria. The crystal violet stain, which is purple, is then added to the bacteria. Iodine is then added to trap the stain in the cell. Alcohol will then be added to wash out the stain. A secondary stain, pink, is usually added to stain the cells. The gram-positive cells will stain purple because their thick cell wall is going to retain the crystal violet stain. It won't allow it to wash off. The gram-negative cells will stain pink because their thin cell wall does not retain the crystal violet stain, at least not any significant amount. And so the pink will predominantly prevail in the coloring spectrum of your eyes. Okay? Does anybody have any questions so far? Okay, eukaryotes. Eukaryotic cells contain compartments that can be bound by internal membranes. The eukaryotic cells, except for plants, by the way, do not have cell walls. Their plasma membranes is a single lipid bilayer. The eukaryotic cells have membranes inside the cell that are going to form internal compartments. These will compartmentalize the cellular functions, many different cellular functions. Some examples will be things like nuclear envelope, endoplasmic reticulum, our mitochondria. Interesting enough, our mitochondria is its own cell to a large extent. And in fact, it dictates many of our functions that we had originally thought were human functions, are actually mitochondrial functions. For example, which do you think actually tells you whether somebody is likely to have bipolar? Is it their human DNA or their mitochondrial DNA? Mitochondrial. Yeah. In fact, most mental diseases are more highly linked with mitochondrial than what we usually call the human DNA. Yeah. So it's kind of funny because there's so little human in our humanity. Yeah. Anyways, what time is it? Okay. So the nuclear envelope is going to be a double membrane. This is going to be connected to the endoplasmic reticulum. And so right here we can actually see the ribosomes. This is the ER. Right here we have the nucleus. This is the nuclear pore complex, of course, and this is our DNA. Okay? And we can see this is going to have the mRNA. We'll assemble it, get it all intron, exon, ready for processing. We'll put it out there. The ribosomes are then going to attach, synthesize our different proteins. Your ERs can gather those up, filter them out, send them to where they need to go, right? Isn't that cool? Fantastic. So the membrane budding and fusion are going to be highly controlled processes. The receptor-mediated endocytosis, is going to be the process by which cells take up molecules from their environment. The receptor is going binding induces membrane invagination by the action of intracellular clathrin and dynamin. And this is actually the process here. These diagrams actually match each of these right here. Can everybody see that nicely? Okay. One of the coolest figures I've ever seen in any biochemistry or biology textbook. Okay, and so this right here actually has the start where we're starting to take the clathrin and we're going to eventually have the clathrin coating. This being the clathrin coated pit right here. And so literally we're taking this, lining it up. This is then buckling it in and creating the start of our structure that's going to suck this in, right? Everybody say that okay? And then the clathrin keeps building up and this has a curvature to it. And so what's happening is it's deepening this and pulling more of that cell membrane bilayer in, right? Okay. And then eventually, this is going to end up with dynamin building up. This will fully constrict and then merge are two sides of the cell membrane. Literally pinching it off, right? And then we get that right there, which is essentially the liposome, right? Depending on purpose, it could be something, have slightly different naming. In this case, vesicle would be the correct name for the specific one we're going to be looking at. The vesicle fusion to your plasma membrane is going to be critical for our neurotransmitter release. The fusion of a vesicle to a membrane is going to be key to the release of the neurotransmitters that'll form the neuron into the synaptic cleft. And so right here we can actually see the vesicles moving up into the synaptic cleft where they're going to do their releases. Again, this is going to be your direction of your nerve impulse. And so here we have synaptic vesicles. This is the presynaptic membrane right here. And again, this is the synaptic cleft itself. These synaptic vesicles are going to merge with this synaptic cleft cell membrane. This then is going to release the signaling molecule into the postsynaptic membrane right here. The area between these postsynaptic membrane and the presynaptic membrane, that being the synaptic cleft. Okay? And then that, typically in most cases, it would be a polarity difference, so there are signaling molecule ones as well, would cause a polarity difference, which then would be detected and propagated through. Okay. So this is the receptor-mediated endocytosis of the transferrin receptor. This will mediate the cellular uptake of iron. So free iron is actually toxic to cells and tightly bound to transferrin in the bloodstream. Have you guys ever heard of soap scum? Soap scum? So if you use soap or detergents where you have very hard water, one of the hardnesses being iron, right? What ends up happening is the polar heads all bind to the iron. The non-polar tails end up exposed to the water, right? And so it falls out and precipitates. This is one of the many reasons that iron is actually quite toxic. The other reason being that it actually reacts with a variety of proteins, carbohydrates, etc., right? So iron's actually a pretty good catalyst, or a really good oxidation reduction reaction substrate. So the complex formation between the transferrin receptor and the iron-bound transferrin is going to initiate the receptor-mediated endocytosis. And so again, here we see the iron-bound transferrin. This is again our clathrin-coated pit here. We see here we have a mechanism, the transferrin receptor here, that can bind this iron-bound transferrin right here is getting bound to. See that? Okay. And so again, the clathrin will pinch these off. We then have in here the acidified endosome right here. And so acid is put in. This causes release of the iron, and we can collect it into this right here. Everybody see that okay? What happens then is once we've collected the iron up, we remove the iron from here. We're going to then merge it back into the outside, and the iron will free transferrin will be released. Okay, and so we end up recycling. Yep, this right here is the iron-bound transferrin. The transferrin being the little blue thing, the iron being the yellows, and so this is going to end up binding to the receptors. And then the receptors are actually going to be what gets pinched off into these endosomes right here. This can go like that. And then what's going to happen is we're going to have acid brought in, and the acid is going to be what causes the iron to release. Okay. It's actually very similar to our diffusion, our column over there that we get the DI water from. In case you're wondering, that works the same way essentially. It attaches to the iron, and then we acidify it to release all the ions at some point. Yeah. Anyways, we then are going to pump the iron out, and then we merge that back out to release the iron-free transferrin, the transferring from the receptor. That makes sense? Okay, snare complexes are going to initiate your membrane fusion. Your snare proteins will be the guided membrane fusion to ensure specificity. They're going to draw the appropriate lipid bilayers together through the formation of tightly coiled 4-helix bundles. These are the 4-helix bundles here. They're largely determine the compartment with which vesicle will fuse. And so you can see here that they basically have the start of the lipid bilayer here for the vesicle to fuse to and for the cell membrane to fuse to, right? And then they have these alpha helix structures to merge the two and cause them to get pulled in. Okay, and that's actually where we're going to stop for this chapter. Does anybody have any questions on any of that? Not so far? Yeah, make sure, do the homework. That'll help you realize if you had missed anything or didn't understand something. Yeah. That does a pretty good job of taking you through your paces, right? I think I covered most.